Yeah, the late Richard Borgens had this made this interesting point where he said that as soon as so super early on, as soon as you have a membrane that segregates some goodies inside the organism versus outside, you're likely to have a charge imbalance. And then if you get injured, so let's say the membrane gets perforated, you immediately get a free vector to the side of the damage because of the short circuit of the electric field. You get and you get that for free. That's a free gift from physics. You don't need a genome. Yes. You don't need right. You don't need pathways. You don't need any of that. Immediately, you know where the damage was. So if you have some sort of electrosensitive subunit, as occurs in wound healing and whatnot, it just the physics tells you where to go to plug up the damage and so on. So I'm guessing there are many of these things that are just, so these free gifts that we get from chemistry and physics, and then the evolution exploits those in different ways.
So since you already mentioned evolution, let's talk about it. So how these kind of systems will evolve?
I think, as I say, for bacterial cells, for single cells, the membrane is what divides them from the outside world, and there's no way, how does it know what's happening outside? Well, things impinge on the membrane and cross that membrane, and the cell with its biochemistry has to figure out, what does that mean and what am I going to do? Should I swim that way, or should I swim this way, or how am I going to behave in response to this? So there's never, it doesn't know that there's an oil slick over there or whatever it may be, but it's picked up something and it needs to make a decision as a cell what it's going to do next. So I quite like the ideas from Carl Friston on this, who talks about a Markov blanket and the cell membrane as being a Markov blanket. And effectively, it's translating between the language of the outside world and the language of biochemistry inside. And that language of biochemistry inside is trying to find this low energy state, in effect, which is its homeostatic preferred place to be. I'm not sure I convey very well what he has to say on this, but the way I interpret it or understand it is, well, if you're in a bad environment, then your proteins start to unravel and denature, and it's that's actually a higher energy state and not a place you want to be. And if you move over there and your proteins start renaturing again, then that's a better place to be. But you still need to integrate all of, you've taken a statistical sampling of the outside world. You've got a correlation that says this happened, therefore do that. But you take multiple samples of this through multiple receptors and they're conflicting. Some of them say go over there, some of them say go over there. So how do you integrate all that information and make a decision about what to do? And that's where the cell membrane, the plasma membrane, if you like, begins to integrate all that information. If you're, if the problem is that you're poisoned and your substrates don't work, then your membrane potential is going to fall. If your membrane potential falls, then you move over there sharpish. So, it provides you with information on your state in the world as a single entity. And I like to think of that as the simplest possible imaginable form of the stream of consciousness, which is to say it's integrating real-time information about your state in relation to the world in terms of, am I good or am I not good? Really, that's the simplest thing. Do I go there? Do I stay here? What, it's that seems to me to be a fundamental decision for a living cell to make, which is based entirely on its need to survive in the world, and which is integrated by the membrane and in effect by electrical charge on the membrane as you integrating that information.
Yeah. That's a really good point. Chris Fields talks about this kind of thing in bacteria as an early form of metacognition, because if you imagine you're a bacterial cell in some sort of gradient, you can measure the actual molecule that you're interested in. But for example, it might be poisonous. There might be a poisonous version of it. And so what you might also want to have, and some bacteria have this, is a way of measuring indirectly instead of tracking the thing you really want to track. What they track is your own metabolism. So right. So it's a kind of a self-monitoring loop, basically a simple example of metacognition where where you say, okay, I'm not going to worry about the details of what it is I'm trying to metabolize. Let's just, how are we doing? Are we like, and that gives you, there's some trade-offs there, but it gives you this incredible power of coarse graining over all kinds of details to have a large scale variable to deal with, right? And metabolic state and membrane potential is a fantastic example of that. And I think getting back to the evolution question, one sort of hypothesis I will put out there is that the fact that we have these metabolic needs and the fact that you don't have all the time in the world to process every possible scenario because you're going to fall apart and be eaten is really, I think, a key driver of intelligence for the following reason. If you are a creature that does not have these constraints, it doesn't have metabolic needs. For example, like a modern AI, which is plugged into the wall, it has all the energy it needs. It never has to worry about getting any energy. If you're a biological agent that arose in evolution under these constraints, that you are constantly under this metabolic pressure, one of the things you cannot afford to do is to be a micro reductionist, meaning that I'm going to track all the micro states in my environment. You do not have time to try to be a Laplacian demon. There's just no time for that. Yes. Instead, there's great pressure to become really good at coarse graining the world into chunks that you can large scale affordances, observables, control knobs, and pay attention to those and completely sort of abstract what's underneath. So that pressure to cut up all the details into higher level things that you're going to represent, like my metabolism, my resting potential, right? These higher level things means that all of the ability to generalize, the ability to abstract from individual instances to general rules and general observations, and even dare I say, for advanced organisms like us, the ability to tell stories about agents doing things. In other words, to have an agential view on the world. In other words, in modern humans, to have a view of free will, as in I am a thing that does things, and there's a tiger, and tigers are agents that do things, and so on. All of that has, I think, its origin all the way at the beginning. The first agential models were made by microbes, who had to adopt that stance. They had to adopt an agential stance in order to survive in real metabolic, under metabolic constraints. And I think what evolution did after that was scale it up to creatures that can then verbalize it, like us, who can say, "Well, I've got free will and this and that." But all of this goes all the way back to the beginning, to the necessity to make models of large scale variables doing things, being important control knobs. If my voltage is off, if my metabolism is off, I need to change my voltage. I'm not talking about this sodium ion versus that sodium ion. I'm talking about getting my voltage fixed up. And that's, that I think is a driver of the ratchet for intelligence.
I mean, a couple of things. One thing about voltage and bacteria, there's a guy called Ken Nielsen who for years has been culturing bacteria on electrodes. And the thing is that lots of bacteria, it's always it's proved impossible to culture them in a pure culture, which is to say just those bacteria and nothing else. And the reason is basically that they dump their electrons onto their neighbor, which is another bacterial cell of a different species, let's say, which wants those electrons. And so it's a syntrophy. They're effectively living alongside each other. But the way to kind of escape from that is you give them an electrode and you set that the potential, the electrical potential of the electrode to, let's say, minus 300 millivolts or something like that. And they either dump their electrons onto that or they take their electrons from that. But then they'll grow on that surface at that voltage. And if you switch the voltage to, say, minus 304 millivolts, you get a different bacterial population growing there, and you change it by another 3 or 4 millivolts, and you get a different, and you effectively grow all of these pure cultures of bacteria by just shifting the voltage by just a few millivolts. It's exquisitely sensitive. The way that again, it's how all this works at the level of cells, I don't really know, but the voltage in your environment, and the way in which where you're offloading your electrons and where you're gaining your electrons from it's fundamental to how cells grow. But coming back to evolution again, bacteria are using their membrane as the interface to the world. But then we have eukaryotic cells that have internalized mitochondria, and they start using the plasma membrane for something different, and they can be now much more interesting in what you do with your plasma membrane. You don't have to be using it for energetics all the time. You can be using it for much more interesting and subtle things, including changing shape and becoming a phagocyte or beating cilia, whatever it may be. But now this kind of simple idea that your metabolism is directly plugged into your membrane and gives you an immediate integrated feedback on your state in the world becomes much more complicated, because a lot of your metabolism is happening in the mitochondria. You may have got scores of mitochondria. They may all be doing different things depending on where they are in the cell. And the cell now has a kind of a bigger problem of how do I integrate all this extra information about how I'm doing in the environment. And it seems to me almost necessary to bring it. I mean, you could imagine that it could all work by phosphorylating everything, but I find it very hard to buy. I find it much easier to imagine that either we've got networks, but more likely we're simply signaling electrically between different mitochondria and between mitochondria and the cell membrane, so that the cell is integrating its state still electrically, but in a more subtle and sophisticated way. So this is a big, I would say, a big challenge at the origin of the eukaryotic cell. How do you rewire yourself to work as, instead of being a consortium of cells that have their own independence, but now some of them are inside, how do you kind of rewire so you act as an entity, as an individual organism at the level of the cell, with all of these things inside, all of which are metabolizing potentially in different ways? So there's a problem with integration there. And then you have the next level up, which is you become multicellular, and then you've got another problem with integration. So you're kind of you're ratcheting up the hardware required to keep a signal which is intelligible. And by the time you got into a central nervous system, it's enormously complex, of course.